My name is Stephen Gordon and I run a pulmonary research lab, and I'm thrilled that the students were able to interact with some of the postdocs and the scientists in my laboratory. We had imagined that art students would have little interest in the work we do, and it turns out to be completely wrong, that actually the art students are intrigued by what we do, and we are thrilled and excited by the way they represent our work. So I personally find the three-dimensional models the most exciting. Other people have found the artwork in the drawing books better, but I think we should do more of this. I think it's terrific, and we have a lot of other scientific ideas that I think the art students would probably enjoy expressing.
I really did think it turned out like this, like exhibition, like having it against the white background, it makes everything stand out. The picture's really effective with the contrasting colours. Really impressed with how it's come together.
It's one of the most unusual and unique exhibitions that I've ever been to. It's more than just art, and it's more than just, I shouldn't use the word just in other thing, it's more than art, and it's more than science. And I'd like to congratulate absolutely everybody involved. Make sure when you go out from this place, you tell people to come in here and look at it.
My name is Gabrielle Del Pronte.
My name is Megan Cox.
And we're both students.
Create a sculpture that looks good and will show other people how a HIV cell works.
Yeah, I also aim to create something that's like aesthetically pleasing and intriguing to the public, so much so that they're going to be able to understand aspects of science that they wouldn't normally consider. I don't take any science, I don't study any science at further level. I'm beginning to understand science in a way that I wouldn't really have thought, because I know that art is used for art therapy and just generally as like an aesthetic, but to use it in science is really making the most of the language and how people feel about art.
Yeah, I think it's really helpful, especially for people that don't have a scientific mind or don't fully understand how the inside of your body works, and especially if they're in hospital and they want to know what's happening to their own body, to see it in a sculpture or in another way might help them to understand better.
My name is Dr. Adam Wright. I work at the Liverpool School of Tropical Medicine. I work for the Biomedical Research Centre, and my role in this project is to provide some of the scientific content to help them interpret some of the research that we're doing in the laboratory on inhaled vaccines in the context of in the context of 3D structures, art structures. So the aim of today is to engage with art students and to communicate our research into the way in which the lung responds to infection and to inhaled vaccines. To communicate this scientific knowledge so that they can have inspiration to build 3D structures, 3D artistic structures in a way that can communicate some of the research that we're doing in the laboratory to the public. I've really enjoyed coming to the schools and engaging with the students about the scientific content of the artistic work. It's allowed me to uncover some knowledge that I want to learn more about in terms of how things interact on a 3D scale, which is not something that we think about a lot in the laboratory. And I'm really looking forward to seeing some of the artistic work that is produced in terms of helping myself, helping my colleagues, helping the way that I think about my own work and about how I communicate that to others. It's been a very positive experience. So I've enjoyed developing that aspect of my communication with the public, and I've also enjoyed seeing and talking and thinking about ways in which cellular interactions on the microscopic scale can be recreated on a more physical setting.
My name is Simon Abrams. I'm a postdoctoral researcher in the BRC. I got involved in the project basically through, I did my undergraduate degree in Liverpool, and then I did my PhD in haematology in Liverpool also. Started to research sepsis with them. The aim of the project really is to translate our scientific knowledge to the art students, and then for them to represent this knowledge through art. So basically it's transferring our scientific knowledge, and then hopefully through this the students can learn a little bit about what we're doing in the BRC, and then through representing this in art they can transfer this knowledge that they've learned to people that see the artwork that they've produced. It's quite hard when you're used to being in a scientific background day in, day out, to come out of that and be able to speak to people that may not be experts in the field and make sure that they're able to understand it. It's sometimes difficult, but on the plus side from that, it makes you understand your own work a little better. But the most enjoyable part has really been to be involved in an arts project. So to actually be able to see the things that we do in the BRC being translated into art, I think it's an amazing thing that we're doing here. And actually I would have liked to have, had a little go at sculpture in myself. I think it's added to the work in the labs, because when say we've been writing our presentations, it makes you think more about the research that you're doing, and when you start thinking about this and ways that make it easier to explain to the students what you're doing, this sort of thing, it helps you understand, I think, the stuff that you're doing better yourself. So I think that's how it's helped me with my work anyway.
So how did I get involved in the project? I was contacted by a colleague and a tutor from my foundation course who knew what I'd gone on to study, which was 3D design, and I've also got a background in conservation. I used to work for NML in ceramic and sculpture and glass conservation. So the elements of my 3D work combined elements of science and the arts as well. So I think that background tends to lend itself to this project. When you're making something or constructing something, you're using physicality, you're using materials, whether that's the science of materials or the science of the body and tissues. There's all kinds of interactions happening in lots of different processes. So I see them as being intertwined anyway. Enjoyed the main thing is, I think it's such a there's so much inspiration initially from the subject matter that we're dealing with. I think there's just such a vast array of shapes and forms and information that we can gather from the subjects given by the scientists. I find that really, really exciting, and there's just so much mileage to exploring that. It's been a huge learning curve, learning obviously about the science of it as well, and also how I try and communicate that in the classroom with students. Trying to not taking for granted my language of 3D as well. My knowledge of the science of the subject has had to develop alongside the students' knowledge as well. I've felt at times I've got to be a little bit ahead on that, so I can pre-plan in terms of materials and where we might be going in terms of developing kind of shapes and forms that we can look at. That initially coming into the realms of science and the initial meetings with the scientists were actually quite daunting, and I feel a lot more confident about that towards the end of the project, because now I can say I see it in terms of shapes and forms and scenarios and textures, and For the scientists, I think similar to me, you know, they're dealing with lay people in terms of, you know, I'm a lay person to them, so they've had to explain to me in lay person's terms their subject, and similarly I've gone back to them with samples and you know, bits of my artwork that I've tried to do in response to their work, and I've had to try and explain to them what I'm trying to explain in 3D. I might be explaining that in terms of spaghetti and pasta, and in terms of what kind of you know, materials I've used to explain maybe a permeable surface or something. But I think having something that's so ordinary and so accessible as a tool for them to explain the science to me, I think that two-way thing has really broken down the barriers and just given us a vehicle of something that was really accessible to just to chat about the subject. So I see that as a huge opportunity for students to really be able to be able to get. I think it's a fantastic project to be involved in, and I just hope that their enthusiasm can really show in the work that they're producing.
My name is Nina Doran. I'm here today as the external evaluator of the project with the Belvedere Academy students. I will be running the focus group with the Belvedere Academy students today, asking them a series of questions about the processes and the science that they've learned, and I will be writing up a written report. How do you feel about trying to explain something in 3D as opposed to maybe writing about it or drawing about it or speaking?
That's the main clot, and then the other side is coming in to make the clot bigger. Because in sepsis, the body has all the normal responses to an infection, but it's uncontrolled.
So why those colours?
I kind of try to stick to like the neutral kind of, obviously with the red and like pinks and things like that, which would be the same kind of things. And then on the outside, I kind of did the blood capillaries, obviously on like the outside of the lungs, with the like blue and red of like oxygenated and deoxygenated.
You can't get that in a textbook, because you remember in a textbook you just have it, you know, you wouldn't think of it as being all cobwebby and nesty and all layered, would you?
Yeah, that's like,
show you layers, but you wouldn't really see it as layers.
Especially like with the blood capillaries and things like that, you just see them as lines, whereas they are actually like fibres.